Okay, so the next part of the assignments will be the last part of MetaPipe, which is basically the functional assignment. Um, so a quick overview of the general sort of overview of functional annotation. If you're doing assembly on metagenomic samples, this is basically the generic workflow for everything. So you're starting off, I mean, on the first or our first day, you did filtering, you did the merging, and then at the end, after assembly, you basically got some contigs out, right? So that's our starting point. So we have assembled contigs. The first step is usually gene prediction. So you want to find out genes. You want to find the genes from the contigs. They can be fragmented, they can be full length, but in the end, you'll have a set of genes. And these genes, you want to annotate them with different kinds of annotations. It could be enzymes, it could be whatever. There's tons of databases available. And in the end, you'll have a set of genes which have some annotation. And then you want to do a functional analysis in order to basically answer the question you have at hand. So back to MetaPipe. This was day 2, not day 1. This is what you did. Today you did this thing. So basically LCA classifier with the silver mode, and then you visualized it in the Corona chart, which you're probably looking at right now. So the last part of today is basically starting this step. So we're going from contigs, gene prediction, and we have 3 different annotations. So InterProScan, you probably heard Rob talk about that in this morning. So that's a collection of 14 different databases. We have enzymes specifically, and there's a blast against UniRef. And then in the end, we'll get the GenBank. But for today, you're only logging into the site and basically submitting your files. Because this step is actually quite computational heavy, you can't do it on a VM or just one machine. You need to have a big computer cluster. So a typical runtime, if you did this with, let's call this a medium metagenomic sample, so 30 gigabytes, would be something like 5 to 6 hours in pre-processing. So that's what you did on day 2. And that's not fiddling around on the computer, that's basically runtime. And then this particular bit would be 1 to 2 days on really big computers. So what we did, since we have 17 groups right now, we don't want to wait for, maybe 30 days, because that's a bit long for this course. So we've reduced this data set, the one Eric just explained, the one with feces from babies. So it's, I don't know, Eric, 2 and 3 contigs? Yeah. So it should be about only 100 genes, but in reality you would have thousands or etc. So let's see. Yeah, so a brief overview. We're using a metagene annotator to predict the genes. So it's meant for metagenomic data. It's a bit old. I think it's from 2008, but it's still performing very good. And from all the reviews of novel or later gene annotation, metagenomic gene annotation programs, the difference between them is so little that it doesn't really matter. As I said, we have a specific tool for annotation of EC numbers or enzymes. We're using UniRef 50 because UniProt-KB is simply just, the default to big database, it's just too big. It would take maybe a week for one big sample. So we've reduced it to a clustered version, which is basically uses a parent sequence of a cluster of UniProt. And then, of course, Interpro, which is this collection of 14 databases. Rob went through this morning. There's domains, conservation, evolutionary relationship, etc. You'll see in Artemis tomorrow how this looks. So basically today it's really short. You navigate to a site where we have our marine reference databases, which Nils mentioned. You can also blast against these databases, but we won't do this. Won't do that. MetaPipe is available through a link, so you basically click MetaPipe. In order to get into the site, there's a password, and it's in your manual. So just before you get to this page, there's a login. And this login is only to the site. So as you can see, when you get there, you're not really logged in. So there's 2 logins. Don't get confused. So in order to actually use MetaPipe, which is the second login, you need to have either an ORCID, a LinkedIn, or a Google account. And I'm guessing at least one of you in each pair have one of these. So basically, most of you probably have the Google account. Click the Google account. You will go through a certain 2 or 3 pages with associate my account with Google, etc. And then there's a form in the end, and it's really basic. You just fill in your names, your email addresses, and then your Google account will be associated with Elixir AI, which is this login. And then when you're done with that, you just navigate back to this site, same address, and now you can see you're logged in, so you have your name in the upper corner. You click Annotate Now, and you'll get to this page, and there's a file on your VM, which is basically the contigs, reduced contigs from the assembly step. Upload them. Don't touch these; they don't do anything at the moment. So this portal is really a beta version. It's not publicly available, so you guys are kind of exclusive alpha users, I guess, to use it. Upload your file, press Start MetaPipe, and then that's basically it for today. Because we will run, it will probably take maybe 6 to 7 hours with 15 or 17 samples running. So tomorrow morning we will continue. And then basically you can go to see your results and you'll get the annotations. There won't be any text here because, as I said, it's still in beta. So you can just press go to results and you'll get an overview of all your files. So basically right now, there's a GenBank file, which is the one which is called export.gb, which is the one we're working with. But also, if you're interested, you'll get your genes in nucleotide and protein format. There's an annotation in JSON format, etc. But I guess this is not interesting for most of you. So basically, you download that file, and then we'll have some fun in Artemis looking at the contigs, the genes on the contigs, and look at some functionality. Basically, browse through all the annotations, and there's some questions. And if you didn't follow what I said right now, all this information is, of course, also in your practical assignment file.